remember. Okay, just makes sense. If you, like I said, if you don't have it like memorized, and I hope you don't, I hope you have it more where it's applied so you know where it is on your body, you can do the action. But when all else fails and you just blank, then just look at the fibers. Where are they going? Follow the direction. And then when you have an insertion, it's always, let's say 98% of the time, going to be distal to the origin. Okay? So if it's on an appendicular skeleton, it's going to be the insertion distal, the origin's going to be proximal. So you can follow the muscle. The one that's closer to the main axis of the body is going to be the origin. Where it's going to attach is going to be the insertion. Look at the model and see what bone it's attached to. There's no need to memorize a bunch of stuff when you've got some helps. Just visually look at it before you start answering, okay? Okay, and so I've listed those for you, and I've also identified them in the same manner in which the chart in the lab manual has categorized it. So head and neck muscles, those are all going to be listed. And then you've got the muscles that move the shoulder, muscles that stabilize the shoulder, so you've got them listed in the same way. So that it's just, for you, a checklist without all the other additional information. Okay, it just cleans it up for you. Now the second handout is one that may help some of you. If you find that it doesn't, just put it aside. I just wanted to make certain to get it to you, just in case it might help. So for example, when some of you are trying to determine what's a synergist or an antagonist outside of appendicular skeleton, okay, let's say we're looking on, the main axis of the body. What we can then do is this particular chart helps you to visualize the action. So if I look at the first item on here, it says sternocleidomastoid, and it flexes the head. Okay, so whenever you're writing an action on the lab test, you do not just write it flexes. Well, it flexes what? Give me the detail. It flexes the head, flexes the arm, flexes the forearm, and I'll put that on the marker board, the erase board, so that it reminds you when you're taking the test, you need to write what it's going to move. Just writing flexes is -1/2. It's got to be complete. Flexes the head. Now the sternocleidomastoid, when they work together, some of you already identified that it will then flex the head. When I take it back to anatomical position, I'm going to contract my trapezius to get it back up. And so we've got to go back for those actions. So we'll do a review on those tonight, just to make certain that you know what those various actions are. Because I can also flex the head, extend the head back to anatomical position, and hyperextend the head. Okay? So we want to be able to then answer a lecture question. Which of the following muscles serves to flex the head? Name a synergist to that, something like that. So you want to be able to then get it, do the action, and then get it back to anatomical position. Okay? Now, when you see on this list, one says that it flexes the head, and the other one says that it extends the head, you should think automatically, those are antagonists. They do opposite actions. Okay? So when you see actions that are similar, like for example, the trapezius and the rhomboids are going to adduct the shoulder. Okay? Those are going to be then synergist to each other because they do the same action. One is a primary, but then one is a secondary. Then I see that, I don't have anything on here under that L base, but I can see serratus anterior and the pectoralis minor are both going to adduct the shoulder. So those are synergists to each other, but they are antagonists to the upper two. Adduction and abduction are opposite actions. That means that they're antagonists. So this may help you as you're studying the actions, okay? Do what it says. Flexes the arm, extends the arm. And then when you feel that muscle on the arm, you should, when it flexes, it shortens, it should get bigger. When it relaxes and extends, okay, then it's going to then be the opposite action, right? And then if I'm going to do that, then the one on the other side of the arm is going to then be the antagonist. Does that kind of make sense? So what I'm trying to do is to really emphasize the importance of making sense of it instead of trying to memorize all this info. I would go nuts if I had to try to not memorize all this stuff, to be honest. But if I could move my body when I'm taking the test and I can go ahead and go, oh yeah, I know where that's at. Hey, makes it easier. And like I said, I'm not going for exotic muscles, I'm going for the large ones and the ones that most likely you're going to be working with when you do any kind of a workout. Okay, so these are the ones that hopefully this helps you. If it doesn't, don't worry about it. You can figure out whichever way you want to learn the material. The bottom line is that you just have to learn the material. So you can see that I can either go crazy and do a lot of physio on the lecture test, which I don't like to do because it's really heavy duty, and kind of balance it out with some of the anatomy. So I will go ahead and have about 2/3 on the physio, about 1/3 to balance it out, adding in then the anatomy of the muscles, right? So that you can, oh, okay, this is gonna, it's not so doggone heavy. Now, I gave you the 3 essay questions, and what you wanna do is work on those over the course of this week, next week, the weekend, whatever, and then I'll let you know before the test which one I'm going to go with, because then that's the one you need to write on, which is what you did with the essay that all of you picked up today. Okay, so that essay is 5 points. You got 1 or 2 ways you can do it. You can go ahead and add the 5 to the 60 because the last test was worth 60 points, right? Mm-hmm. Make it 65. Add the number of points that you got for the essay to the raw score, and that then becomes out of 65. Or go ahead and leave it at 60, and then just go ahead and then do whatever out of 5. Doesn't really matter. It is on the lecture side though. The essay is lecture material. So it goes on that side of the grade tower, okay? On this one here, I don't think I got 5 out of 5 on it. Because it was, there's a thing that those of you who got 4.5, you were right there. Either you missed the cell, calcitonin targets the osteoblasts to then take the calcium out of blood circulation so that you're building bone, putting it in. And then the parathyroid hormone is then going to come from parathyroid gland, and it targets the osteoclasts, right? So the one that was most often missed on that was the osteoblast. You said calcitonin, and you said it takes the calcium out of blood, but how does it do that? So that was that one little part that I was missing on that section there. And then the other thing that was missed was some of you had the hormone. You didn't tell me where it was coming from. Maybe you did, but you didn't tell me what happened to the other hormone. Calcitonin goes up, parathyroid goes down, and then vice versa. That's why I said which one is going to stimulate and which one's going to inhibit. So that means you had to have both of those hormones in order, and I added that on there to make certain that you got that done, okay? So I felt that everybody got at least a 1. Okay, because there are 4 parts to this thing, so I just, okay, that's fine. But those that, I mean, that one little part, okay, that 1/2 point came from usually that osteoblast for the calcitonin. Okay, but otherwise, some of you write very well, very pleased. Okay, it made sense. Okay, so I appreciated that. Appreciated that. Okay. All right, so I'm going to go ahead and finish out the molecular mechanisms tonight. And before I start, I just wanted to make a little bit of a mention. Okay, I'm sure that most of you have been seeing what's been going on with college campuses, university campuses in terms of the protests, pro-Palestinian protests, etc. You might have seen them on the news, okay? And to be honest, isn't that what college is about? I get very upset when they say you cannot demonstrate. They're not being violent. Now, if you start throwing things, then you can get in trouble. But if you're standing there and you're doing a peaceful demonstration, that's the right and the critical thinking ability of college students to say, you know what, this is wrong, I'm gonna take a stand. But you don't have to be violent, okay? And it's reminiscent, I mean, I'm sure you've talked to your grandparents, because I'm probably the same age as some of your grandparents, okay? I'm a product of the 60s, and mid to late 60s. I graduated in 72. So that means I was part of the feminist movement, and you know darn well I demonstrated on that one. I participated in Roe v. Wade. I had a friend who died from a botched abortion, so no way. You choose what you want. I'm not going to legislate morality to anybody. You make up your own mind what you want to do, and that's you, with you, and your own moral beliefs, ethical beliefs. But other than that, don't tell me what I'm going to do with my body. Right? I was also part of the demonstration for anti-war for Vietnam, because that was it. I have friends that were drafted to go to Vietnam, a war that everybody wasn't even a war, that everybody just gave everybody who came back from Vietnam the absolute worst grief I've ever seen in my life. And they did not choose to go. I had friends who just simply said, you know what, to hell with this, I'm going to Canada. And they did. But there were those that said, you know what, they got the bad luck of the draw. So they were chosen, they were drafted to go in. You have to demonstrate against that. There are certain principles that you've got to demonstrate against peacefully. I was part of the civil rights movement. There was a beginning of busing. Okay, and to tell you, I lived in this inland empire. I came to San Bernardino in 1964. My dad retired from the military at Norton. So I went to San Gorgonio High School. Okay? San Gorgonio High School. That's my alma mater there. And I'll tell you what, when they had demonstrations against that, it was so violent that I had helicopters, I still recall. They had helicopters that dropped 55-gallon drums of tear gas in the middle of the quad to disperse people. Students would have to stay in the room, and then my crazy physics teacher, first he hated women anyway, so the guy was a nutcase to begin with. So we had 3 women in there, 3 girls in there out of the bunch of guys in there, and his son was from West Point, and so he just felt like women should not be in physics. And I'm going, oh my gosh, this is really. So he would, try to take points away from us all the time. Stinking candy wrapper on the ground, minus 10 points off of a test. I'm going, this guy's really retarded. Anyway, excuse my language on that. But bottom line is that he would just simply make us come in, and he had a cattle prod. And if anybody came through the door, he would just give the cattle prod. That's what I grew up in. I grew up in the first Earth Day. I was an environmentalist too, because at that time DDT was killing all the brown pelicans in California, so I had to work with that too. I demonstrated all the stinking time, because you know what? Somebody had to say something. Otherwise things would not change. So to me, a non-violent protest and demonstration is part and parcel of the democracy of this country. So I'm glad that they're out there being peaceful as much as possible. They have no idea what in the world, oh yeah, just go ahead and arrest me. I don't think they understand fully, but it doesn't really matter. The bottom line is that they're making a stand. And to me, that tells me they're thinking. Nonviolent protest is what we have to do. And this kind of is reminiscent of the 60s and what I grew up in. The ability, only at that time, only about 30% of medical school applicants were females. Women just were not allowed to go into the sciences. So you have to make a stand sometime. So I applaud the demonstrations that are peaceful. There's no need to get violent with this, but you need to make a stand and say, you know what, this is wrong, if that's what you believe. And then make it observed, and then do what you gotta do. You gotta get arrested, you get arrested. You know what I'm saying? But I'm hoping that, awakening some of you. The Roe v. Wade is a big thing for me right now too. I mean, there's a lot. It's like, are we going backwards? Are we going into like, we're going to follow the constitutional law from 1864 in terms of abortion rights? 1864. You have slave owners. Why in the world would they let you have an abortion? And we were trying to follow that? So, I mean, so to me, I don't care whether you have it or not. You have the opportunity to make the choice, and that's inherent in every female that's going to be carrying a baby, whether or not. I'm not a I hate the word pro-choice or pro-life. Yeah, I'm for life. What the heck, man? I'm a biologist. What do you want? But on the same token, don't call me pro-choice just because I think that women should have the right to make their own decision. So sometimes the circumstances make you think, really, what do I believe? Really, what should I stand for? What is the right thing to do? So I just wanted to make a little bit of a commentary and, say USC is not going to have a graduation. Well, my feeling is too bad, so sad. I don't know what else to say. It's almost like COVID. We didn't have graduation then, did we? No. Did we die from that? No. We just didn't do it. Maybe it will come back later. Maybe it won't. My poor grandson never had a senior prom, never had a senior graduation from MLK, and King High, nothing. Life happens. So as much as I feel sorry for people who aren't able to do things like that, well, you know what? It's the sign of the times. It's a sign of the times is what it is. And it's kind of like a huge change going again. So we have to think about things like that. So I just have to say something, because I don't want you to not think we are in college. This is where you finally can say something if you want to say something. Okay, now, let's go back to our little microfilms. Okay, so on this diagram, all right, so can you tell, why do we call it a thick filament versus a thin filament? Why do we not just call it actin and myosin? What's a thick filament defined as? It's a bundle of myosin, right? It's not one myosin molecule with the two parts to it. It's 300 of them together. That's one thick filament. When I say thin filament, what are my components of a thin filament? Actin, tropomyosin, and troponin. That's a thin filament. So actin and myosin are just simply the proteins. The structure, thin filament versus thick filament, refers to then an actual organization. So on a lecture test, which of the following best describes a thick filament? A myosin molecule, a bundle of myosin molecules, okay? We're going to get specific. Tell me, is this relaxed or is this a contracted sarcomere? And how can you tell?
Relax.
Okay, it's relaxed. Why? I see an H zone. Right? I see an H zone. This diagram does not illustrate an M line. That's not the purpose. All this is doing is showing you the relationship of actin and myosin in a sarcomere. Okay? So I can tell that this one is a relaxed because I see a distance representing my H zone. So I can translate that into a lecture question, couldn't I? Okay, you can figure out how I'm going to ask that. All right, so this is then a relaxed sarcomere. And these sarcomeres, as we've indicated earlier, are found along the length of a myofilament filament, but it's more obvious. The striations come from what you observe in a myofibril, right? So in the model that we saw in lab, you've got to be able to identify the myofibril, the myofilaments within, and then the striations are due to then the periodic cycle of the A band versus the I band, right? Okay, so this kind of reviews all of that for you. The one thing I do want to point out, because this is going to be part of the information that I'm going to be lecturing on. So if I look out here, this portion outermost that surrounds the entire structure, this is a cell. That's going to be my sarcolemma. My sarcolemma is going to dip down into that myofiber, and it's going to be in the form of a T-tubule. So T-tubule is equal to phospholipid bilayer, isn't it? Yeah, it's the sarcolemma coming in. Now we learned at the end of that unit 1 that my cell membrane has a polarity. It's got a membrane potential, doesn't it? And there are two really important cell types where the membrane potential is very, very important in terms of how it works. That would be in a myofiber and in a neuron. Okay? So if you recall, an action potential, okay, let me go back. A resting membrane potential, RMP. A resting membrane potential is when I have the ratio of 3 sodium to 2 potassium across the membrane, right? And I have to, in order to maintain that, I'm going to have pumps, those sodium-potassium gates that are going to help clean it up so that I have a membrane at rest. If I open a gate, then sodium rushes in and it causes then the membrane to react. That reaction is the beginning of an action potential. So it goes from rest to action. And I have to have that happen in order to ultimately contract an entire sarcomere. I've got to disrupt the resting membrane potential in order to initiate the contraction of the myofiber. Okay? So it's got to go from rest, doing nothing, like relaxed, to then becoming active and contract. Now we already learned what ion it is that's necessary for the muscles to contract. All of you put that down on your first essay question. What's my ion?
Calcium.
Calcium. So now I've got a little bit of a connection. Don't know the whole thing yet, but I know that calcium's got to come in, and it's going to attach onto the troponin, because I already said, oh, the troponin is where calcium attaches, and that's going to initiate then a movement of the tropomyosin away from the active site in order for then contraction to occur. Okay? And in order to get the calcium, which is stored in the myofiber, into the sarcoplasm, I have to open up the gates and let the calcium out. Where was that calcium stored? Terminal cisternae was where I'm going to have the greatest abundance as part of my SR, right? So that's where it's going to be. So that means that I have to have an action potential coming in, which is going to trigger the gates to open the terminal cisternae, calcium comes out. When it comes out, it can then bind to the troponin. Did that kind of make sense a little bit? Okay? That's what the gist of tonight's lecture is. How does that all happen? In the end of that lab 16, there are a series of plates that kind of describe that sliding filament theory. It shows you that. And then in addition, it has the excitation contraction coupling. We call it the neuromuscular junction. So those two are static. But like I said, if you go to YouTube and just put into the search engine sliding filament theory, then you can actually see the animation. And maybe some of you have already done that. Okay, do the same thing at the neuromuscular junction. Okay, so you can see what kind of activities occur. And then convert the animation into a verbal description. And then convert that into a written description, because that's what the essay question is, isn't it? Describe an action potential. Describe the sliding filament theory. Describe the neuromuscular junction, excitation, contraction, coupling. I have another one in there that's the energetics, which is next week's information. Okay, so I have then my sarcolemma, and it comes into the myofiber as in the form of a T-tubule. Okay, so that can undergo an action potential. Okay, so that's what the SR is. We're okay with that. We've already gone through that, and hopefully everybody's comfortable with that bit of information. Okay, so the T-tubules, and I asked you to look at the myofiber model to see where that T-tubule aligns with. Is it on an A band? Is it on an I band? Where is it? Okay, so you want to be able to describe what you saw on that myofiber model as to where the terminal cisternae and the T-tubule, the triad, where is it located? On top of what part of that sarcomere? Okay, because then that will give you a little bit of a better understanding, a visual, on how we get the calcium from the terminal cisternae to the troponin. Okay. So here you can see on a highlighted part, this is then an extension of the sarcolemma, and you can see that it comes down in the form of a T-tubule. You'll notice that that T-tubule is linked by way of proteins to the terminal cisternae. These are gates. That when the voltage comes down, it opens the gate. And that then allows for these little red dots, which is calcium, to then rush out into the sarcoplasm. Why? Simple diffusion. Area of highest concentration to the area of lower concentration. And as it rushes out, it then attaches onto my troponin. Attaches onto the troponin, moves the tropomyosin out of the way of the active site, and then that allows then for the myosin heads to attach onto the actin dimers. Okay, so you have that. All right, so here it is. We kind of finished out our lecture with this. You can see then that the myosin heads are slightly bent, okay, but they're not attached yet. They're slightly bent, and that's due to the presence of ATP. Okay, so when the ATP is there, remember what I said about the myosin heads? It's a globular protein. Globular proteins are typically going to be enzymes. There's an active site. The substrate for that active site is ATP. So when ATP is hooked onto that myosin head, it's going to cock it a bit. It's going to bend it. Okay, when it actually breaks the ATP into ADP and phosphate, okay, inorganic phosphate, then that's going to trigger it to actually cup. Okay, so it actually bends and moves. So what we're doing is moving the actin molecules over the myosin. The myosin's held stationary. Why? What keeps the myosin in position? The M line. So those are the myomesin molecules, the proteins that hold it in position. So the myosin doesn't move. It's the actin that moves, and it slides over the myosin. It's ratcheting. I'm just ratcheting it, okay? And it then shortens the distance between the two Z lines in a sarcomere. And that then is going to initiate the contraction in the myofiber. How does that equate to the muscle? Well, that's next week. We're going to talk about how does that convert into then muscle contraction. But we've got to look at the molecular level. So the myosin heads are slightly cocked by the ATP attaching onto the that's the substrate, onto the enzyme. So then when I have the myosin heads in close proximity, what I'm going to do is I have to have, in order for me to do that, I'm going to have calcium. I got to remove the block. The regulatory molecule for contraction is tropomyosin. It regulates it because it hides the active site. The troponin's hooked onto the tropomyosin, so when the calcium attaches onto that, it can then move. Okay, so the calcium is the trigger for the contraction. Okay. So can you describe then how we're going to have that contraction at the level of a sarcomere? What ions are necessary? Okay, and you might even want to put in, where's the sodium and potassium? What if I say sodium and potassium? Where is that even associated with anything? Okay, that has to do then with the resting membrane potential of the sarcolemma. And in order for me to cause a stimulus to come down, I'm going to have to shift it out of balance, and then it can go down into the T-tubule. Okay? Okay, so then what happens is that once I have the tropomyosin moved, the myosin heads can attach directly, and then I'm going to trigger my ATP, I'm going to break it, and it's going to cuff it. And that's all I'm going to do. I'm just doing this. I'm just moving that actin. Come down, attach, bend up again, attach, move it. Come down, bend it, attach, move. That's all you're doing. That's all. It's just cranking it. That's all it's doing. It's just cranking it. So then ultimately, that sarcomere is going to contract. Now this one here is not the this is still relaxed, right? Okay? That's still relaxed, but what I'm going to do is ultimately this. So what happens to the A band? What happens to the I band? What happens to the H zone? What happens to the distance between the Z lines? What happens for every one of those in terms of that contraction? And you've got in your textbook, there's a paragraph that actually goes through every part of that, and it just 1, 2, 3, 4, 5 tells you exactly what's going to happen. So if you kind of get lost in me describing it, go back to the reading because it's very clear. Again, that's the reason why I like Mira. She writes very well, makes it very simplified. Okay, so everybody okay with that so far? So that's the sliding filament. We're sliding the actin over the myosin. Okay, we need ATP. You saw the ATP there. We need calcium, right? And now we're going to find out, well, how does that all start? How can we know that skeletal muscle is under voluntary control, so I've got to tell my muscle to contract. So how do I get the message down? Okay, question.
Why is it called a theory and not like a theorem?
Well, whether it's a theory or a theorem, it's law. It's law versus theory, okay? So when it's a law, that means that they haven't found anything that is opposite to that, right? So until they do, and chances are they're not, but we don't know everything, we're going to call it a theory. Sliding filament theory. This is how we explain everything on how we have muscle contraction. But we could throw in some other kinds of chemicals that we haven't yet developed that can paralyze a certain part of that step and say, oh no, that's not really what happens. It's this. Okay? So you've got then that theory that kind of helps to explain how a sarcomere contracts. So in order for a skeletal muscle to contract, I have to have an impulse coming from the nervous system. Okay? So I have to have then a portion of the nervous system sending out a message to my myofibers, because why my myofibers? Well, my sarcomeres are there. The sarcomeres contract. So I got to get it down to the myofiber in order to get that sarcomere to contract. So here is again a cross section of the spinal cord. We're going to find out there's a difference between this white matter and this kind of the little butterfly, whatever you want to call it, that's gray matter. We're going to find out that the cell body of the neuron is located in the gray matter, and then it's going to come out in the white matter as a nerve. Okay, and then that nerve will then have a nerve ending. The nerve ending is actually the axon terminal. I'm going to go into some more detail on that in just a moment. So every one of these myofibers has an axon terminal. One axon terminal, one myofiber. Okay, but you can see that at this clustering of that axon terminal, I've got multiple branches. That's why I have junctional folds. So on the myofiber, you found the junctional folds, so now you can see. Oh, that's one part, okay? I can then have multiple areas where I have those junctional folds. And you can see then, we call those axon terminals a motor unit. So a motor unit is going to innervate, it's an axon terminal, from a spinal nerve that's going to innervate a myofiber. Where is that spinal nerve exiting? Through my intervertebral foramen. So it's going out of my spinal cord, okay, from my spinal cord, out through the intervertebral foramen to finally make its way to the muscle. So motor units are the axon terminals, the branches, that are then going to innervate the myofibers. And I can have, on a motor unit, multiple myofibers. So I've got one message going to 4 myofibers. Or I've got one message going to 100. Okay? So I can have multiple branching. The bigger the muscle, the more the branches. The more refined the action, the fewer the branches. So if I look at this motor unit here, I follow it, you can see it's going to here, and it's going to here. So it's innervating 2 myofibers. On this one here, it's going to this one, and this one. It's going to 3. The point that that's making is that you can have multiple myofibers stimulated from a single motor unit. The bigger the motor unit, the more branching. So let's make sense of this. If I'm going to do any kind of extension with my quads, okay, that's a big group of muscles right down here in my legs. I want many of the myofibers being stimulated by a single motor unit. So let's say I've got 400 of my myofibers that are going to be innervated by that particular motor unit. I'm going to get from one message 400 responses. However, I'm playing the piano. And on the piano, I'm only using a certain number of muscles that are in between my little digits here. I want refined action. So I'm going to have fewer myofibers that are going to be innervated versus a blinking of an eye. I usually have 4. So the fewer the number of myofibers to a motor unit, the smaller and the more detailed the work has got to be done by that particular muscle. Okay. We're going to say that again when we can look at the muscle. So you'll hear it one more time, not from this perspective at the molecular and cellular, but as an organ level. So here we have then these little axon terminals. Now this is a highlighted of one of those little branches. There's my axon terminal. There's my axon. So on that myofiber, you recall seeing that yellow cord? That's my axon. Okay? So the nerve information is coming down that axon. And as it comes down, it will then be in close proximity to the junctional folds, but does not touch them directly. There's a space. So I found here, this is my neuron, axon terminal, and can you believe that this axon terminal, the membrane on that is called an axolemma? It's an axon, membrane, axolemma. My junctional folds, sarcolemma, associated with the muscle fiber. And then I have this gap in between. We call that the synaptic cleft. So that's why I have on your list the cleft or gap, and then you have then the junctional fold, okay? Now if you notice, the junctional folds, why do we have folds of anything? What's the reason for that? There's just one basic reason for that.
Increase area.
The which one?
Surface area.
Increase the surface area. Anytime I fold any part of it, I'm increasing the surface area to get the activity. So aligning my gut, I'm going to have lots of folds. Folds on top of other folds. Just so that I can maximize the uptake of nutrients. So I'm going to have here the junctional folds, our sarcolemma, And on the sarcolemma are receptors for the chemicals that are going to be released from this axon terminal. The greater the folds, the greater the number of receptors, the greater the probability of attaching and causing then a reaction. Okay, so here's my axon terminal. Notice that my axon terminal has mitochondria. Okay, so in my model, on my myofiber, I can actually see some of the mitochondria that are in my axon terminal. And then you see these little dots. Now, these dots are actually vesicles. And in the vesicle is a chemical. We call it a neurotransmitter, generically. Okay, nerves never touch a muscle. There's always a space. So that means then in order to activate that membrane, the receiving membrane, I'm going to have to release a neurotransmitter. It transmits the nervous impulse from the axon to the target. So this vesicle, because it's associated with the axon terminal, is called a synaptic vesicle. When I have a axolemma, a synaptic cleft, and a junctional fold, that complex of three structures, again, axon terminal, synaptic gap, sarcolemma, that's called a synapse. Membrane, space, membrane. It could be membrane of a neuron, space to another neuron. It could be a neuron, space, muscle, okay? So whatever the target might be, there's never a direct connection. So because this is called a synapse, so what's in a synapse? The membrane of the neuron, the gap, and then the membrane of the receiving cell. In this case, it's myofiber. Okay? It could be neuron, gap, neuron. It could be neuron, gap, muscle, myofiber. So I have then the synapse, and I'm going to have then in that area, in the axon terminal, synaptic vesicles, which then contain the neurotransmitter. Neurotransmitters are proteins. So the proteins can be made way up in the cell body, which is here, and then can be sent down all the way down here, or you have a little mini factory down here. I can make it down here if I need to. So anything that's made, I don't want that neuron to use it up. I'm going to package it and get ready to send it out when I need to. So these are synaptic vesicles containing the neurotransmitter acetylcholine. So for this particular transmission of information, the neurotransmitter is acetylcholine. And we abbreviate it as A, capital C, small letter H, acetylcholine. Another example of a neurotransmitter that you might be familiar with might be serotonin. Maybe dopamine. Those are all neurotransmitters that are going to do a specific action on the receiving membrane. Okay, so these blue dots that you see here in the cleft or gap, and I don't care if you call it a gap or cleft, it doesn't matter. Those are my acetylcholine molecules, and they're going to attach onto a receptor. Okay, so we've got a little bit of an idea. So once we have a receptor, once we have a neurotransmitter attaching onto the receptor on the sarcolemma, it's going to cause a shift in the membrane potential. It goes from resting to now activated. So we initiate an action potential. Typically what we're going to find along a membrane, remember we've got lots more sodium outside than potassium inside. So the outside of the membrane is going to be positively charged. the inside of the membrane is going to be negatively charged. So when you see this configuration, it tells you, hey, that membrane's at rest. Positive outside, negative inside. When I shift it, and I have a negative outside and positive inside, we've now stimulated that membrane, and we initiate what is called an action potential. So that's your first essay question. Describe an action potential. So what we do is we disturb the resting membrane potential, open the gates, sodium rushes in, and so then we shift the charge across the membrane. That is equivalent to now activating that particular cell, okay, that cell or cell membrane. Okay, now, this action potential is going to be propagated. So let's talk about how that happens. So actin is made up of individual G-actin subunits, those are the little dimers, they're globular, with active sites that bind myosin heads. Remember, they've got the little active sites. Myosin heads can attach for muscle contraction. Muscle contraction occurs when the calcium ions attach onto the troponin, but they're released from the terminal cisternae, right? So we know this, okay? So how does this all happen? I'm kind of giving you a little bit of a preview. Now let's go ahead and go into a little bit more detail. Okay, so as I said, I have then, in this case, a motor unit, another motor unit. This motor unit, you can see, is going to innervate 2 myofibers. This one here is going to go 1, 2, 3. And it's just hypothetical. Is that the numbers that always happen? No. It's just trying to show you that we have then this association. It can be anywhere from 4 to several hundred, okay, of those myofibers from a single unit. So again, just showing you that motor unit 2 is a larger motor unit. It's going to innervate 3 myofibers versus motor unit 1, which is in comparison smaller. It only is going to activate 2 myofibers. All right. When that axon reaches that sarcolemma, it's going to look like this. This is then my sarcolemma. Okay, I can kind of we want to get ourselves oriented here. And I have then these 3 axon terminals. Okay, so I can have multiple branches coming to that one area. And you can see that I'm going to talk a little bit about this later, but this is myelin. And it's not going to be at this end. At this point here, I have to have exposed membranes. So this is the exposed axolemma to the exposed sarcolemma. Does that make sense? So my axon terminal, this is a cell, isn't it? As weird as it looks, it's a cell. So it means it's got a cell membrane. But because it's associated with the axon, we call it an axolemma. This myofiber doesn't look like a typical animal cell, but it's a cell. So that means that that membrane is called a sarcolemma. So I'm getting now detailed on the kind of membrane that I have. So I have then the axon terminal interfacing with sarcolemma, but there's no direct contact. Okay? There's a gap. All right, so, and again, you should be able to, as I list them, can you find this? Can you see my myofibrillin here? Can you tell on the diagram what my myofilaments are? Can you tell on the diagram where my sarcoplasm would be? Can you tell on here where my A band is? Can you tell on the diagram where my I band is? Can you tell on here where my Z line is? Can you tell on here where my N lines is? Okay? So that's the same thing as on the myofiber. Not on the sarcomere, but on the myofiber, right? Okay, so go back and review that if that was a little confusing. This is what it looks like on an electron micrograph. So you see the axon terminals going to the individual myofibers. Okay? So what we're talking about, just to kind of back up, because we're kind of getting so much into the detail, what in the world are we talking about? How does the information get down to the myofiber to contract? To ultimately to the sarcomere to contract? That's what we're looking at. how does that information relay event occur? Okay, so here we have again, this is what you saw on the previous diagram. The axon terminals, which are branches off the axon, coming in close proximity to the sarcolemma. Okay, I can see in here now more clearly my synaptic vesicles, and I can see that on my model, so I'm going to ask you, hey, name that structure. Tell me the name of the chemical that's in there, because now we now know it's acetylcholine. So I then have my synaptic cleft, and I have then my junctional folds. Okay, and the old-fashioned term is motor end plate, so when you see it in the lab manual as motor end plate, don't get yourself confused. It's just talking about, oh, the junctional folds, okay? So we have then this configuration. Now notice something bizarre here. Hey, in order for me to release my acetylcholine, calcium's got to come back into my axon terminal. So that means I have calcium channels in here, and I'm going to have calcium come in. Remember, you wrote that down too, as another function associated with calcium, nerve impulse transmission. So when the calcium comes in, it causes then the vesicles to migrate to do exocytosis. I gotta have calcium coming in for that to happen. So then these vesicles are going to migrate toward the periphery, and it's going to, by way of exocytosis, release the acetylcholine, which can then bind to the receptors on the sarcolemma. Okay, that makes sense so far? Okay. I know I'm going kind of slow, but I just want to make certain that everybody's on the same page. Okay, so now, name the chemical that activates the sarcolemma. Acetylcholine. That's what activates it. That's what opens my sodium, potassium, my channels, so that the sodium rushes in, it starts the action potential. The acetylcholine is going to be the important neurotransmitter that when it releases and attaches onto the sarcolemma responds by undergoing depolarization. So when that happens, I'm going to attach that acetylcholine onto the receptor. It then is going to open the channels, and it's going to allow for sodium to rush in. The first part of that activation is called depolarization. So I'm going to depolarize my sarcolemma. Now once I have that attached, then everything goes within milliseconds. So then I'm going to have then a contraction of the sarcomere. But I don't necessarily want to have the muscle contract for an hour. I want, I got to lift, relax. I lifted that pound box of sugar, I put it down on the counter, I'm done. I don't want it to continue to stay in that position. So in that little gap, there is an enzyme called acetylcholinesterase. Remember the ASE ending tells you it's an enzyme? So the acetylcholinesterase will take all of the unattached acetylcholine, break it down into an acetyl group and a choline group, and bring it back up into the axon terminal. Then I have mitochondria, which produce ATP, that can then hook the two components together, repackage it, and store it back in another synaptic vesicle. I don't want to keep making acetylcholine. If I can avoid it, I'm going to recycle. So my acetylcholinesterase is going to take the unblocked or the unused acetylcholine, split it, and bring it back into the axon terminal. Okay? So that acetylcholinesterase is going to stop the contraction from continuing. Okay? So if I have, let's say I have a block to my acetylcholinesterase. I put in a synthetic antibody to it. Let's just say that. And it attaches onto my acetylcholinesterase. What is the outcome if I block that enzyme? It keeps contracting, right? Okay? That contraction, continued contraction over a long period of time is called tetany. And it's the outcome of when you have tetanus, where you have prolonged contraction of muscles because the acetylcholinesterase has been blocked. So when you have tetanus and you haven't been immunized for it, and you get tetanus, you're going to have a toxin that blocks acetylcholinesterase, and long-term effects, you're going to have continued contraction. I've seen photographs of patients where their entire back, you know, you can only do so much, you know, there's hyperextension, okay? I've seen it where it's almost like almost 90 degrees with continued contraction as a result of having tetanus, okay? So I can block that and then have pathology. Normally, everything works fine. I'm going to stop the contraction. My acetylcholinesterase is going to be activated. I'm going to break down the acetylcholine, and I'm going to bring it back up into the terminal, axon terminal, and I'm going to repackage it. Okay? So the question would be, which of the following conditions is a result of continued or lack of acetylcholinesterase stimulation? Continued contraction of a muscle. Okay. Now, what if I block the receptors? If I block the receptors to acetylcholine, then they can't what? They can't contract. That's what we do with Botox. We're injecting a toxin that blocks the acetylcholine so that we get rid of the wrinkles. You're given botulism toxin. That's why some people are now coming down for some reason. People are going to these Botox parties and it's not a pure product, and so people are coming down with botulism as a result of the Botox. Okay? It's derived from botulism, the Clostridium botulinum, which causes botulism, but I'm blocking. They can't contract, so it gets rid of my wrinkles. Okay? Just relaxes everything. Okay? So I can have some pathologies if I block a receptor or I block an enzyme, okay, that breaks something down. So again, in order for this sarcolemma to be stimulated, the acetylcholine must attach onto the receptor, causing then a change in the membrane potential and initiating an action potential, a depolarization event. Okay? So then that depolarization is then going to continue down. Okay? It's going to continue down the entire, it's like throwing a little pebble into a pond. The ripple effect is going to go all over and then go down to T-tubule into the myofiber. So I will then have an initiate this. This is first going to be the localized starting point. Then it's going to make its way down a T-tubule. When it goes down the T-tubule, we already saw that there were other proteins that were attached, so they're going to open up the terminal cisternae. In order to start the whole thing, I got to have calcium. So now we've got, where's calcium important in my diagram? Where's sodium important? And where's potassium important? So you've got multiple places where you have to have calcium in terms of muscle contraction. Here, the entry of calcium into the axon terminal. Right? And then, once I get over here, I'm going to have to attach the acetylcholine onto this, and then I'm going to initiate an action potential down here, and ultimately, where's my other calcium store? Terminal cisternae. So then I have to open up the terminal cisternae and release the calcium. And then where's that calcium gonna go? To the troponin, right? When it attaches onto the troponin, what molecule moves out of the way? Tropomyosin moves out of the way. It opens then the active site. What molecule then attaches into the active site? The myosin head. Okay. Okay. So it gets heavy duty with different ions. Okay. So the voltage, in order for this to happen, the voltage, this calcium comes in. It then activates the synaptic vesicles to release the acetylcholine. So calcium initiates that. Which of the following ions initiates the release of acetylcholine? Calcium. Okay. The acetylcholine diffuses across the synaptic cleft and attaches onto the receptors on the junctional folds, which is then going to cause an action potential. So what ion enters when we initiate an action potential from resting to now depolarization? Sodium. Okay, so the action potential is initiated in the muscle. That allows the sodium to enter the cell. Okay, and now we're going from resting membrane, and I'm going to put on the test as RMP, resting membrane potential, and then it's going to depolarize the membrane. So that means I'm going to reverse the charge from outside being positive to negative and vice versa. Okay, so it's going to happen here. Okay, and then in order to stop any further contraction, I'm going to have to activate my acetylcholinesterase. Okay, and where is that found? Synaptic cleft. And what is its substrate? Acetylcholine. Okay. And what is its product? Acetyl group and choline group.
What activates the enzyme acetylcholine?
What initiates the acetylcholinesterase? Okay, let's say that you have stopped giving the information down to the muscle. You've lifted your 1 pound box and you put it down, and your brain is saying, okay, we're done. So information is sent down to that, and so then that's when we start activate. No more contraction, no more sodium, no more calcium going in. And then that acetylcholinesterase is going to start active on. And it's only within a milliseconds, so it doesn't take that long, unless I'm setting more and more input. If I'm lifting a 50-pound weight, I'm trying to do some resistance training, that's going to be different than just doing a 1 pound. Okay, so the neuromuscular junction. So what is a neuromuscular junction? NMJ. Just break it down. Neuro, my neuron axon terminal. Muscular, sarcolemma, junction, where they come together at the cleft. They're not touching, but that neuromuscular junction is comprised of axolemma, a sarcolemma, and a synaptic cleft. Okay? And in order for information to be then sent down to the muscle to contract, I'm going to have to release acetylcholine. So that's my neurotransmitter. Okay? And then that acetylcholine is stimulated when I have then the gates open for calcium coming in. So that's what's going to initiate it. Migrates exocytosis. And then it's going to go into the synaptic cleft and ultimately attach onto the axon terminal. Onto the sarcolemma. Okay? Okay. So the last part of this, I just want to make certain that we all understand what we're talking about with an action potential. We saw this before in Unit 1 when we were talking about a membrane at rest. And the ones that we're talking about are the ones that are more irritable. Every membrane has an action, a resting membrane potential. But it's much more pronounced and much more dramatically different when we're looking at muscle cells because they're irritable and neurons because they're irritable. That allows them to conduct information. So when we're looking at then a resting membrane potential, this is what it would look like. We're looking at two ions in particular. We're looking at sodium versus potassium. Now this diagram shows it very simplified, and that's all we need to know for right now, okay? But the reason for the negative in here, remember, where are my peripheral proteins? On the inside face of that membrane, and most of my peripheral proteins are negatively charged. So I've got sodium on the outside, positively charged, and the peripheral proteins are on the inside, so that's why it makes it a dramatic difference in the muscle cell or in the myofiber. So the inside face is negative. That's rest. Then, when I start shifting, okay, I'm going to, first part of that is called depolarization. And what I'm going to do then is open the gates, and I'm going to then exchange ions. And it will continue in a segment. This portion, then this portion, then this portion, and this portion. It's very much like a wave when you're doing the wave in a stadium. Okay, it's gonna undergo a change and a shift. The first part of that action potential is called depolarization. So in depolarization, sodium rushes in. Okay, that's the first part of an action potential. And then when we reach a certain amount of sodium coming in, I'm going to initiate an action. I gotta have background noise. You know, when you're sitting here and listening to my lecture, you don't hear the buzz from the fluorescent lights. You don't hear the fans going. But when I stop talking, can you hear them? Oh yeah. Because what we did was we stopped that particular initiation. So we stopped that. In order to initiate an action potential, I've got to reach what is called a threshold. That's kind of like the background. Once I get over the background, then I'm going to do something about it. Okay? So I have to have then a threshold value. We're going to talk about that next week. Then I'm going to have depolarization, where I'm shifting. And then once I reach the maximum shift, it can't go any more positively charged, or I've got to stop and bring it back down and get it back to resting. So at the peak, that's the peak of depolarization. Then I've got to repolarize it. I've got to get it back down so that I can receive a second message. Got to clean it back up. Remember, it's like the wave. So I do one segment, clean it back up, go to the next segment. See, so it's going down sequentially down that particular membrane. This is what it's going to look like, and this is a diagram in the back of your lab manual. This is what it looks like. So you can see then excitation and contraction coupling. So here is my axon. Release of the acetylcholine attaches onto the receptor. Here I am at rest. Nothing's happening, right? How do I know nothing's happening? It's positive on the outside, negative on the inside. That's resting membrane. So this is at rest. Everything is nice and happy and nothing's really happening. And then once I start having a shift, it's going to go down along the T-tubule. It's going to activate the proteins here, open up the terminal cisternae, and then the calcium is going to attach onto the troponin. That's what we call excitation, exciting this part here. Contraction, sliding filament here. Excitation, contraction, coupling. That's what ECP is. That's what's on the essay. To describe then the excitation, contraction, coupling. It's getting from a resting membrane potential due to the release of the acetylcholine, and then propagating that down the T-tubule to the point where I then open up the terminal cisternae to release the calcium. Okay? So I'm going to go ahead and, once the action potential starts, once I reach a threshold, there's no stopping it. It's called an all or none. It's got to go to the end. Okay? So we have to, first of all, get to that threshold where it's gonna trigger. And if we never reach it, it's gonna go back down anyway. But once I get to the threshold and it fires, there's no turning back. It's gonna go, we call it an all or none phenomenon. Okay, and then we have to get it back to resting membrane in order for another stimulus to come down and activate it. So we'll go ahead and stop here, and we'll pick up the rest of this. So that means that my quiz on Tuesday will have 20 questions worth 10 points, half a point each. It has everything from the first slide up through this one here, to repolarization. Okay? Okay. I need you to get that done because next week is even busier than this. So we gotta do it kind of like in chunks so that we're comprehending. All right, so let's go ahead and let's start lab at, let's see, 6:20, 6:40. Come on back in.